Everyone's quieted down, so I guess
Might as well.
we should start. It's 3:30. It's my pleasure to introduce Dr. Jen Wisecaver, who came to us all the way from across the street in the Biochemistry building
Long walk.
to give a seminar. Jen did her bachelor's degree in biology at Humboldt State University in Northern California. She then went on to the University of Arizona for a PhD in ecology and evolution, where she started studying algae. From there, she went to do a postdoc at Vanderbilt University, which is in Nashville, is that in Nashville, Tennessee?
Yep.
Where she studied under Antonis Rokas.
Yep.
And she started doing fungal and plant specialized metabolites and the evolution of the biochemical pathways that lead to the production of these secondary metabolites, specialized metabolites. And then she came to Purdue in 2017 as one of the Center for Plant Biology hires. And she is now an associate professor in the biochemistry department. And she also has a courtesy appointment in botany and plant pathology department as well. So go ahead, Jen.
All right, cool. Well, thank you. I love having a captive audience to listen to me talk about algae and Prymnesium parvum for about 50-ish minutes. So thank you for being here. So what you can see on the left side of the screen is the species that I work with, Prymnesium parvum. You can see Prymnesium here, phagocytosing up a bit of cell debris from a former green alga. And it's this feeding behavior. I'll be talking a little bit more about this later on in the talk. And when Prymnesium gets to sufficient densities in nature, you can see it can get quite dense. The toxin that Prymnesium produces can be toxic to gill-breathing organisms in the environment. And you can see here a massive fish kill that has resulted from a Prymnesium toxic algal bloom. And you may have been noticing some more news recently about toxic algal blooms all around us. And so I figured I'd highlight a few of the most extreme examples. If we're looking within the Great Lakes, the toxic algae that is the biggest problem, particularly in Lake Erie, is the cyanobacteria microcystis. And you can see a bloom of microcystis from 2011, a record-setting algal bloom actually from space. And unfortunately, this bloom isn't really record-setting anymore. Subsequent blooms year after year have been just as bad, if not worse. The city of Toledo actually in 2014 lost its drinking water for several days because of a buildup of the toxin that microcystis produces that was in its drinking water supply. And so nobody was allowed to drink water from their water supply in Toledo for at least 3 days. And so this just keeps getting worse and worse. Florida is a state with a lot of problems with toxic algal blooms. One of the worst, most devastating species is Corynea brevis, which is a dinoflagellate. And you can see here another record-setting bloom for this species in 2019 off the coast. And you can see it's called red tide for a reason. And this bloom resulted in thousands of tons of dead marine animals, $2.7 billion estimated in lost revenue. And actually this toxin that Corynea produces in the surf, it can kind of become aerosolized. And so you don't even have to be in the water to be affected. You can just be walking along the beach or standing at this lifeguard station and need to wear a respirator because their toxins can be cause skin and respiratory irritation. And then the species I study had its own unprecedented ecologically devastating bloom in 2022 in the Oder River of Germany and Poland. And you can see here the effects of this Prymnesium parvum bloom. It affected essentially the entire river system, 125 miles of river with Prymnesium parvum bloom, 250 tons of dead fish. And in the aftermath reviews of the bloom, people describe this bloom as being just only a matter of time, because the Oder River is now permanently saline, and that is a condition that Prymnesium requires. It cannot grow in purely fresh water. It needs a little salinity. And so that is the condition that the Oder River is now in due to drought and runoff from agriculture and mining. And Prymnesium is a problem not just in Europe and Germany and Poland, but it's also a problem here in the US. The first recorded fish kill from Prymnesium was in 1985 in Texas. And since then, blooms of this species have been documented in 23 states and counting. And the Great Lakes Aquatic Non-Indigenous Species Information System lists Prymnesium parvum as a species of great concern. So it has the potential for a high environmental and socioeconomic impact if it ever is introduced or gains a foothold in the Great Lakes area. So I pulled off the internet a few infographics, one from the Clean Water Action Fund, one from the Surfrider Foundation, talking about some of these abiotic factors that are associated with these increased incidence and severity of algal blooms. And these are things that I've already mentioned, or at least alluded to: increased agricultural runoff, temperature, increased salinity due to drought and low river flow. And so you can look at all of these factors and see that we have incidents of toxic algal blooms occurring and increasing in severity. And you might come to kind of the conclusion that these are relatively simple systems, that you just increase the amount of nutrients in the environment, you increase the salinity, and you're going to get an algal bloom. And while models that focus on these abiotic factors are very accurate at predicting how algal species will respond to changes in these abiotic factors, there's still a temporal and spatial component to toxic algal blooms that we don't fully understand. And we are still struggling to predict exactly when and where these blooms will occur. There'll be conditions that seem perfectly ideal for a toxic algal bloom, and we won't see a bloom. Or we'll see conditions that would otherwise be kind of at the lower range of what we would expect its species to tolerate, and it will bloom in these conditions. So there's a little bit of extra complexity that we still don't understand. Add to that, we don't understand why some algal species seem to be so prone or commonly forming blooms, while other species, related species, never form blooms at all. So to try to unpack some of this extra complexity for understanding algal blooms, I want to actually step back a minute and actually ask an even broader question, which is, what even are algae? What are these, when we say an algal bloom, what sorts of organisms are we talking about here? And so I have a tree of existing life showing bacteria, archaea, and eukaryotes on the left. And if you're talking about an algal bloom, some of species that are a lineage of organisms that you may be referring to could be cyanobacteria. These are also called blue-green algae. They include things like microcystis from that Great Lake example. And so these are bacteria. So they have smaller, streamlined genomes. They have asexual reproduction. They're still an incredibly diverse group of organisms, but they are a single lineage of bacteria. And if you contrast that with the the eukaryotic algae, like the Karenia and the species I studied, Prymnesium, these organisms are secondarily photosynthetic. They have acquired the ability to photosynthesize in their evolutionary history through the engulfment and endosymbiosis with a free living cyanobacteria that then became an endosymbiont and eventually became a reduced organelle, photosynthetic organelle. But unlike cyanobacteria, which we can refer to as one lineage of organisms, these eukaryotic algae are not monophyletic. They are not a single group of organisms. They're much more complicated than that. And so to explain why, we have to also zoom in on the eukaryotic tree of life And this is our best understanding of eukaryotic diversity circa 2019. And I'm labeling here all lineages of eukaryotes that have chloroplasts, that are photosynthetic. And so everyone in this room, I'm sure, is well familiar with the archaeplastids that contain the green algae and their overgrown land plants in this green clade here. But you can see that scattered around the tree are other lineages of eukaryotes that are also photosynthetic, also have chloroplasts, but they are not related to these archaeplastids. And the way these algae have evolved the ability to photosynthesize is through additional rounds of endosymbiosis. So the primary endosymbiotic event between a ancestral eukaryote and a cyanobacteria gave rise to those archaeplastids, the red and green algae and glaucophyte algae. Secondary endosymbiosis involved the red or green algae now acting as the endosymbiont for another round of endosymbiosis. So we have kind of like a nesting doll situation going on here. And this multiple rounds of secondary plastid endosymbiosis gave rise to these additional lineages of photosynthetic eukaryotes, including the euglenids, the chloracneophytes. They both have green algal endosymbionts or green algal chloroplasts. And then the haptophytes, which include Prymnesium, the species I study, straminopiles, cryptophytes, and dinoflagellates. So dinoflagellates include that perennial species in Florida that I was talking about earlier. And so now when we look at all of this eukaryotic diversity and all of this algal diversity, it's really, these aren't one lineage of eukaryotes. They're actually a bunch of extremely diverse lineages of eukaryotes. And when we're looking at haptophytes or dinophytes, Prymnesium, the species that I study, it is as equally closely related to like our model plant genomes, Arabidopsis, or our model green alga Chlamydomonas. It's no closely related to those genomes than it is to any other group of eukaryotic life. So they're incredibly distantly related. And if we look at kind of traits of across all these different algal lineages, it's unsurprisingly incredibly diverse. They have their genome size kind of ranges the gamut from the smallest, some of the smallest eukaryotic genomes to some of the absolute biggest. They have complex sexual life cycles. Many of them can be multicellular. They have complicated ecologies and behaviors and metabolism, etc. And so one cool example, I think, that really illustrates how different these algae are. They don't represent kind of primitive single-celled plants. These are diverse lineages of eukaryotes that are different from plants. And one of the ways in which they are very different is this propensity for mixotrophy. So that's the capacity for both growth via phototrophy, autotrophic growth, as well as through heterotrophy. So either phagotrophy of engulfing organic particles, or osmotrophy, so taking up dissolved organic nutrients in the environment. So all of these lineages of eukaryotes many species in these lineages are mixotrophic. So that is a very common ecological strategy of algae. And so Prymnesium is no exception. Here, all of the golden cells are Prymnesium, and there's one poor green alga right there that is going to get bumped around by these other cells, by Prymnesium parvum, and eventually other cells of Prymnesium will recognize that there's food there, and they will swarm this poor former green alga, and that cell will lyse, and they will all start phagocytosing up bits of cell debris, like what you saw at the beginning of the talk. Prymnesium and all haptophytes have this third appendage. They have two flagella. They have a third appendage called a haptonema. And I just love this quote from Ralph Lewin. He is a phycologist. He's considered the father of green algal genetics. Look him up on Wikipedia. He's a very cool guy. I love this quote because it really illustrates kind of our bias or maybe the assumptions that are really easy to make when we're looking at these microbial algae. And he says from this paper from 1992 that people had postulated that haptonema might be involved in feeding, but many of us discounted this idea because haptophytes are generally photosynthetic, and little did we suspect what dark deeds could be done by such little plants. I just love that quote. And so I think that if we're trying to consider what might be some of the biological aspects to Prymnesium parvum that makes it so good at forming toxic algal blooms, one of the things we have to keep in mind is how good of a mixotroph it is. In addition to its mixotrophy, it is also very tolerant of lots of different abiotic factors in its environment. So it can tolerate a range of salinities all the way from almost pure fresh water, but not quite, up to 35 PSU, which is the salinity of just normal seawater. And it can tolerate a range of temperatures from 2 degrees Celsius all the way up to 32. So this flexibility and being able to tolerate a range of different environments, I think, plays a role in Prymnesium's success as a bloom-forming species. Another thing that is important for Prymnesium's success is its toxin that it produces. And so this is Prymnesin. It's actually A-type Prymnesin that is 91 carbons long. It is a toxic metabolite that is produced by Prymnesium, and it's one of the largest non-polymeric carbon chain molecules found in nature. It is, and if you look, Prymnesium actually produces a range of different types of these Prymnesins. There's also B-type Prymnesins that are a little smaller, 85 carbons long, and C-type Prymnesins are 83. Although the structure for C-type Prymnesins hasn't been worked out yet. And interestingly, when people did some preliminary work looking at the ribosomal barcoding genes for different strains of Prymnesium, they noticed that the different strains that produce different types of Prymnesins formed monophyletic groups. So all the A-type Prymnesin producers clustered together, all the B-types clustered together, and all the C-types clustered together. And so right away, that was a strong hint that we may be working with some additional bit of hidden diversity that we weren't fully appreciating when we were looking at this, what we considered one species of Prymnesium parvum. We look at it under the microscope, and based on its morphology, we classify it as Prymnesium parvum, but these different clades of Prymnesium parvum are sufficiently diverse that they're all making slightly unique. They're making their own unique types of toxins. So already hinting that we probably have a cryptic species complex that we're working with here. Prymnesium also has lots of additional phenotypic diversity in addition to the types of toxins it makes. It's variable. Different strains are variable in how phagotrophic they are. So if we look at this plot here, we're looking at 4 different strains of algae, of Prymnesium, and the strains where the lines go above 0, that's indicating that those strains are growing faster when given prey to eat. So you give them prey, their growth rate goes up. Versus 12B1, the light blue at the bottom, it's always below that 0 line. So it's actually seeing the addition of prey species in its co-culture as more of competition. It's growing slower in that case than if you just let it grow by itself in a monoculture. And somewhat relatedly, if we look at those same strains, 12B1, the autotrophic one, seem to have a lower average motility compared to the other strains that seem to be more prone to phagotrophy. We also see variation in just their average growth rate. So if you look at 6 different strains of Prymnesium, this is from kind of hard to see data from 1998, but it's interesting because they look at growth rate as well as changes in salinity. And you can see that not only do the different strains have different average growth rates, but that they are responding differently. They have different kind of reaction norms, preferring different salinity levels depending on the strain. So some interesting genotype by environment interactions going on here. Similarly, total toxicity, just average toxicity of the different strains is variable, and also you see variation within strains if you vary abiotic factors like salinity, as well as temperature, pH, and irradiance. So questions that we are interested in studying in my lab is what are the genetic differences that underlie all of this phenotypic diversity that we see in this Prymnesium parvum morphospecies, and how does the genotype impact other aspects of its biology, so including its ability to produce those Prymnesium toxins as well as form those massive toxic blooms. So in order to try to get at those questions, we needed some genome level data for this species. Before we started working on the project, there was no genome level data available for Prymnesium, so we needed to get some reference genomes. And when looking for what strains to sequence, immediately UTEX 2797 stood out as a really good candidate. It's used in a lot of previous studies on Prymnesium parvum biology, its growth and physiology. And so we wanted to include it. It's from Texas, where strains of blooms of Prymnesium are most severe in the US. So it was an ideal or obvious first choice. And so if we look, doing some preliminary analyses, we estimated its total DNA content for this strain and found it to be just a little bit less than your average Arabidopsis 2C diploid nucleus, so about 274 megabases, which is definitely within the range of feasibility, easily not too big a genome to assemble these days. However, one thing that did give us pause was the really high amount of heterozygosity that we observed in this strain when we did some preliminary Illumina short read sequencing. And the high heterozygosity is shown right here. This second peak in this Kamer frequency plot is telling us that UTEX 2797 is at least a diploid, has at least 2 copies of its genome, and that there is a lot of differences between the 2 copies of its genome, such that the amount of differences would probably make it difficult to assemble. It's hard to assemble a really heterozygous diploid genome. So that kind of gave us pause. And while we were looking at it, we were screening some additional strains, and 12B1 stood out as another good candidate. It's another strain from Texas, and its DNA content was a little bit less than half the size of UTEX 2797, and we found no observable heterozygosity at all. So that told us that 12B1 is likely a haploid strain, 1 copy of its genome, to UTEX's highly heterozygous diploid. And so that would be a great one for working with getting a reference genome. But this was kind of intriguing. We wanted to know what was going on there. So we decided to actually, what the heck, we'll sequence both. So we decided to sequence both of these strains using a combination of Illumina, Oxford Nanopore, and high-C sequencing technology. And I don't have time to go into the details of our assembly and annotation pipeline, but I have to call out Bob Auber and Amanda Pendleton in my lab, who both led the effort here on sequencing and assembling and annotating these genomes. And so on the left, we have all of the assembly scaffolds for 12B1, and you can see that it was assembled into 94 megabases on just 34 scaffolds. So really contiguous, nice looking assemblies. And the triangles indicate telomeric repeats. And so you can see that on most of our scaffolds, we have telomeres on either both or at least one end of the scaffold, which indicating that we're probably working with whole chromosomes or chromosome arms in cases where we only have one telomere. And we had 23,800 or so protein coding genes. If you compare that to what we see with the UTEX 2797, we basically see double everything. Twice the genome size, twice the number of scaffolds, and twice the number of predicted procoding genes. So again, really further reinforcing just how heterozygous those two genome copies are in UTEX 2797, that rather than collapsing portions of its genome that are similar to each other and then having the parts where there's differences in separate contexts, we basically were able to fully assemble the two separate subgenomes from UTEX 2797. So very heterozygous, cannot emphasize enough. And so that was really interesting. It suggested that there may be something cool going on in the evolutionary history of this strain. But in order to investigate this further, we needed more phylogenetic context. So we needed more genomes. So this time we went out and picked some representative strains from across the entire species. So we got 4 more A-type Prymnesium producers. I don't know if I remember to say this, but UTEX 2797 and 12B1 both are A-type producers. And we got 4 B-type Prymnesium producers and 5 C-types. And this time we didn't go do the whole shebang with all the long read and scaffolding. We only got Illumina short read data for these additional strains. But that was, so they're much more fragmented assemblies, but it was good enough to get comprehensive gene level data for all of these strains. And so we could use that to construct a maximum likelihood species phylogeny. And so that's what you're seeing on the left here. We have 2,699 single copy genes that went into building that phylogeny. And you can see that we recovered the monophyletic origin of the A, B, and C type Prymnesium producers, which is what we expected based on that barcoding, the ribosomal preliminary data that I mentioned previously. If we look at the substitution rates and compare and contrast between and within the different Prymnesium producing types, we see that there is an increased level of substitution when you're comparing between the A, B, and C type clades, especially when you're comparing with the A and the B's to the C's, you see a much higher rate of synonymous substitution. You also see an increased rate of synonymous substitution within the C type clade, which we thought was interesting, particularly comparing this K0252 strain from Australia to these other strains that I believe are all from Europe. Pretty sure. And so was sufficient increased substitution within the C-type clade that we decided to kind of split them out, suggested that there may be some additional kind of cryptic speciation going on within these C-type producers. And so we split them up into C1, C2, and C3 clades. And the last thing to point out from this species tree is this long branch. It may not look like much, but it is kind of compared to these other little branches. The branch that UTEX 2797 is on is really big. And so that was interesting to us, and we tried to dig into it further. And we discovered that it's on a long branch because its placement was inconsistent across those 2,000 some genes. It was grouping in different places in the tree, depending on the gene. And so here's an example of what I mean. Here's one gene family that went into that species tree analysis. And you see UTEX 2797 grouping with 12B1, which that's another strain from Texas. And it's also grouping with CCMP 3037, which is a strain from Wyoming. And if you look at another gene tree, that went into our analysis, you see Butex 2797 grouping in a completely different location. It's now grouping with RCC 3707 from England and CCNP 2941 from Russia. So it's grouping with these two European strains in our analysis. And so we could kind of quantify that and say, how frequently do we see those two types of phylogenetic signals, those two topologies, across the whole genome? And so we can call the two strains from the US the A1 type clade, and the two strains from Europe as the A2 type clade. And if you look at the Butex 2797 genome, 21 scaffolds grouped with clear phylogenetic placement within the A1 clade, and 31 scaffolds had genes on it that grouped clearly with the A2 clade. And then only 4 scaffolds were phylogenetically uncertain. So with this information, you could say pretty conclusively that Butex 27 is actually a hybrid species, that it has two phylogenetically distinct A-type subgenomes. And that's why it was so heterozygous and why we're able to fully assemble it, its two separate subgenomes. Just to summarize all of the genome diversity that we kind of recovered when we were comparing across all 15 strains, I have a kind of a summary phylogeny or model for all of the strains. And we have our 3 different C clades. We have our A1 and our A2 clades and our A-type hybrids that include the Butex 2797. And we actually found doesn't appear to be these hybrid strains don't appear to be a one-off. We found additional strains that had that same high heterozygosity and appear to be hybrids as well. So 12A1 is another strain from Texas, actually isolated from the same bloom as 12B1, that appears to be a hybrid strain. If we look at the estimated haploid genome size of all of these different strains, we see a huge variation, with the A-type strains having the smallest haploid genome size of around 120 megabases, and then the B-type strains having the largest, but there's a lot of variation within the Bs, as well as a lot of variation within the different C-type clades as well. It's just crazy that there would be that amount of variation in total DNA content, total genome size between what we considered to be one species, clearly multiple different species. And then also, if you look at the ploidy level, we see a lot of variation there as well. So I mentioned 12B1, the haploid, one copy of its genome. But other strains in our analysis appear to be just regular old diploids, not hybrids, but 2 copies of their genome, and then a couple appear to be tetraploid, 4 copies of its genome. And so all of this taken together is suggestive of really considerable genetic variation, suggestive of at least 3 separate cryptic species, but I think probably closer to 7 if we split the Cs up and we split the A types up into separate species. Then we're getting up to 7 cryptic species in this species complex. And so the next big question that we're kind of actively working on, I don't have an answer for you right now, is what's the ecological consequence of all of this? We can now now that we have this evolutionary framework and a better understanding of these different strains, we can go back and revisit some of that phenotypic data that I mentioned, and maybe expand upon it and make some more targeted comparisons between and within these cryptic species. And so now, for example, if you recall the graph showing the difference in the kind of growth strategy preference between mixotrophy or autotrophic growth, now I can tell you that those top 3 strains that grew faster when you give them prey to eat, those are all hybrids. And I can tell you that the one at the bottom that preferred to grow by itself without prey is 12B1, which is one of our A-type haploid strain. So interesting. What does it mean? I don't know yet, because we have to kind of scale it up and look at more strains. But now that we have the phylogeny, we can start to do that. Another question we wanted to answer, that I can answer for you today, is do cryptic species co-occur within a water or within a bloom? We wanted to address this. We wanted to look at this using a survey of recent Texas blooms from 2018 to 2020. We got water samples from Texas Parks and Wildlife Department that we were able to isolate new cultures of Prymnesium. And so I'm showing you the 9 different water samples that we were able to get isolates from. And you can see that there's a range, these samples, there's a range of Prymnesium parvum density in the initial sample from 9,000 cells per mil upwards to 20,000 cells per mil. There's variation in the amount of toxicity present of that water sample. If we co-culture the water, if we expose for trout larvae to this water sample, how toxic is this water to the trout? It ranged from low to high. And then some of these samples were Prymnesium dominant and some were mixed algal communities. And from all of this, we were able to pull out 34 new Prymnesium parvum isolates, all from Texas, all from these recent blooms. So this is something that Nathan Watervert in my lab has been working on. He sequenced the Gatalumina data for all these additional strains and put them in the context of our species tree. And so here in bold are all of the additional strains that we acquired from those recent blooms. And you see a couple of things right off the bat. One of which is that 8 out of 9 water samples had B-type Prymnesiums in it, which is now all of these strains in purple down here. And that is interesting because it's the first documented case of B-types being present in inland US waters. So previously, all known strains of Prymnesium are A-type Prymnesium producers, like our 12B1 and our UTEX 2797. So that's new, that's interesting. And then, and you can see that also, they kind of cluster separately. All of the Texas clusters, all of the Texas isolates cluster with each other. They're not grouping down here with our European strains that we already had in culture in the lab, so that's good. Didn't accidentally cross-contaminate or anything. They do seem to be their own separate lineage within the B types. And I want to also draw your attention to the strains that I've highlighted in red. So these are all from one water sample, 150 ml sample of water from 2020, the Possum Kingdom Reservoir. And from that one sample, we were able to isolate one A1 type up at the very top there, grouping with 12B1, several A-type hybrids similar to UTEX 2797, and then several B-type diploid strains as well. So all from one bloom. So we definitely have these cryptic species of Prymnesium parvum inhabiting the same bodies of water and co-occurring within the same bloom. And so hopefully I've convinced you that these Prymnesium parvum blooms are not these giant monocultures of simple abiotic or simple autotrophically growing, asexually reproducing cells. These are complex assemblages, and we see different strains of Prymnesium and different cryptic species having a different, showing different growth strategy preferences. And then another thing that I haven't talked about yet also plays a role is that other species in the environment show variation to susceptibility to Prymnesium parvum. So if you look at Denaliella and Phaeodactylum, 2 other types of algae here, if you co-culture them with Prymnesium parvum and you start them out with just 1% of the culture is Prymnesium, 99% of the culture are these other species. Within 200 hours, they will all be dead and Prymnesium will have killed them all. This here is how they would have grown if there was no Prymnesium killing them. Versus Prymnesium, doesn't matter if the culture is 50-50 split. Prymnesium parvum, Prymnesium, it doesn't care. it grows fine. It's not, it is resistant to Prymnesium somehow. And so that definitely is going to play a role when it comes to understanding the kind of the community ecology and the kind of ecology and evolutionary interactions going on within these complex bloom assemblages. And so one of the things that we really want to address next is what are the relative abundances? We know that there are cryptic species of Prymnesium parvum present in the same bloom, but we don't know their relative abundances. We just knew that we were able to isolate them from a water sample. What we need is metagenomic data next to see how do the relative abundances of these different cryptic species, as well as the relative abundances of these community members, how are these abundances changing over time, both before, during, and after a bloom event. So stay tuned for that. That is something that we are actively working on in the lab. Okay, so with my last 10 minutes or so, I want to tell you a little bit about a completely separate story. Still involves Prymnesium parvum and its genome, but rather than looking at the ecology of the organism, we're going to look at the biochemistry and its metabolism. So we want to know, now that we have these fancy, well-assembled genomes, can we identify any of the genes that are involved in making the prymnesium, that crazy molecule that I showed you earlier in the talk? And so just to kind of set the stage, if we look at all the different all the different strains that we've analyzed thus far, and we look at presence absence of different gene families in these different strains, we see that a huge number of gene families are variable. The 44% of genes present in a Prymnesium or in the Prymnesium parvum pan genome are present in some strains and absent in others. And if we look at what types of gene families are in that accessory portion of the genome, one type of gene stands out as, functional category stands out as being particularly interesting, and that's these type 1 polypeptide structures, because those are the types of metabolites that, these are the sorts of genes and enzymes that we think would be involved in making that giant Prymnesium molecule. And so we wanted to see, can we find, what do type 1 polypeptide synthases look like in Prymnesium, and can we find any that we think are a good fit for making that Prymnesium molecule? In order to sort of set the stage for that, I have to first tell you what these polypeptide synthase genes look like in other organisms. And so I have an example of some polypeptide synthase genes in Streptomyces, a bacteria that makes this erythromycin. These genes make erythromycin an antibiotic. And so here we're looking at 3 polypeptide synthase genes, VEB, S1, S2, and S3. And these are pretty complex looking genes. Each gene consists of multiple biosynthetic modules, and each module consists of multiple protein domains. And these protein domains are very repetitive. So you see KS, ketosynthase domain, KR, ketoreductase domain, ATP, acyl carrier protein. And so you see them showing up over and over again. And so these are the protein domains that are involved in growing this carbon backbone for this polypeptide product. And so that's an example of a kind of your typical modular type 1 polypeptide synthase. And so we looked for these sorts of genes in our 12B1, in our Prymnesium parvum 12B1 assembly. And we found instances where we saw these protein domains, but the gene models weren't great. For one thing, they're very repetitive. And so those repetitive regions are really hard to accurately predict, call well in eukaryotic genomes, because we want to first mask out these repetitive regions because we think they might be involved in transposable elements or selfish elements. So these things are often getting masked before we even get to gene calling, because the high throughput gene calling software thinks that they're repetitive elements and are kind of discarding this. And so really, There was no way around it. Our collaborator, Tim Fallon, a postdoc in Brad Moore's lab at UCSD, had to manually annotate regions of the genome that he thought might contain these polyketide synthase genes. He couldn't trust our high throughput gene annotations. He had to go in by hand and try to annotate these genes. And what he found when he was done, took a lot of work, but what he found was actually pretty shocking. He found, among other hotspots, I'm going to focus on two PKS hotspots in the genome that Tim identified, and he named these hotspots PKZilla, PKZilla1, and PKZilla2, because the hotspots are 137 kb long. The PKZilla2 is 93 kb long. And what you're seeing are all of the protein domains in one of these PKZillas. So there's hundreds of protein domains present, dozens of biosynthetic modules, all kind of indicative of these PKS synthases, but just enormous. These are actually, and so he got transcriptome evidence and proteome evidence, and long story short, these hotspots look to be single protein coding genes. Probably, I think they are the longest predicted protein coding genes currently known. And so check out the bioarchives paper, he just posted it. And so, yeah, let's compare PKZilla 1 and 2. The order of the modules and protein domains matches the expected biosynthesis for Prymnesium. So I'm not a biochemist, but Brad Moore and Tim are, and they think that it is highly likely that these two genes work together to make the Prymnesium backbone for producing this Prymnesium toxin, which is really cool. Let's compare them to erythromycin. The example of another polyketide synthase or another polyketide product from before. Erythromycin encoded by 3 genes, 8 modules, 29 protein domains. Prymnesium, if PKZilla 1 and 2 really are responsible for making the majority of its backbone, these PKZilla 1 and 2, 2 genes, 58 modules, 239 protein coding domains split over two genes, which really still kind of blows my mind. And just kind of showing you the relative size. This is erythromycin PKS 1, 2. There are 3 PKSs here, and their total coding sequence length and amino acid length versus the PKZillas. So where are we going from here? So there's lots of work to do in this area now. First of all, we have to, these 2 genes are just going to give us the backbone for the Prymnesium product. There's lots of tailoring enzymes that still need to be identified. And so we're working on seeing if we can use our gene expression data to pull out some good candidates for that. We need to compare these PKZilla genes that we see in A-type Prymnesium producers to the B and C-type Prymnesiums, because they have different carbon backbones. We would expect some variation at these loci. So we need to investigate that more thoroughly. And then the other thing I think is cool is just kind of recognizing, now that we kind of know that this sort of enormous gene can exist, I'm kind of curious to go through and look for evidence of more PKZilla-like gene structures in other eukaryotic algae, because Prymnesium isn't the only giant polyketide product that different algae can make. Carenia, that species of dinoflagellate that in Florida, it makes a toxin called brevetoxin that is made in very similar fashion to Prymnesium. And so looking for genes like PKZilla for the production of these other massive polyketide products in algae, I think is something that I'm excited to do, because I think now we have to look for a menu. There is no way any automated gene annotation pipeline is going to identify of a protein coding gene that is 137 kb long. So they could just be sitting there in published genomes and they've been missed, just like they were missed in our assembly until Tim went and looked for them by hand. So I'm really excited to kind of see where this project goes. So hopefully at this point I have convinced you that algae are cool, that they're not just primitive, simple little unicellular plants, but by diverse lineages of organisms in their own right, and that Prymnesium, parvum specifically, is incredibly genetically diverse, but that we are still working on figuring out the ecological consequence of all of that genetic diversity that we see in this cryptic species complex. And so you'll have to invite me to give a talk in a couple of years and I can update you on what we find out. So with that, I'm happy to take any questions. And actually, let me first thank all the students and postdocs in my lab and our collaborators. Asterisks are people whose data I shared with you today. And you can check out both the genome paper and Tim's Prymnesium preprint if you're interested. Now I'm happy to take any questions.
What defines a species in the group? Just the sequence, or is there anything else?
So if I understood the sexual cycle of Prymnesium, I could do we could try seeing if there were any reproductive barriers between, especially like within the A. I think that if you look at how different their genomes are between the A, Bs, and Cs, I just don't think those genomes are so different that I feel like it's unlikely that they could possibly pair correctly during meiosis. Those feel different. They're making different products. But when we look like within the C types or within the A types, we have the A1 and the A2 lineage. They seem to form stable hybrid. We haven't found those hybrid strains thus far don't seem capable of undergoing meiosis. We haven't seen that yet. So it kind of makes me feel like the A1 and the A2 are sufficiently segregated to be potentially their own biological species. But right now I'm kind of just drawing, I'm looking at their synonymous amount of synonymous substitutions in their genome, just looking at how different they are and kind of drawing some arbitrary lines. But I do think that I want to understand the cycle. I think that would be useful information to pair with that.
Can you date them?
I don't know how. If somebody has an idea for how to do it, I would like to hear.
How many variables?
Yeah. Right, yeah. So if we did some work on measuring rates of mutation, I don't know. Yeah, so I've thought about it. I've done some thinking like, oh, if we could date this would be very cool. Because yeah, when did the hybridization occur? Right. I'd like to do all those things. I haven't figured out how to do them yet, though. If anybody has any ideas, let me know. Yeah.
I have a few questions. First is, so what type of protein is encoded by this giant in more than 100 ppm? And does it encode a very large flexible protein with multiple domains, or it is actually involved some alternative splicing or codon species that is with partial splicing?
So let me go back. The proteins, it's going to be just a super big version of this. So they do have some introns. They're small, and I don't think Tim saw any evidence of alternative splicing. But so what they are, they're repetitive modules of the same sorts of biosynthetic these domains that are responsible for kind of growing this backbone. And so that's why there's so many repeated over and over, is they're just kind of growing, they're modular, growing this molecule. So yeah, they don't have much disordered proteins, domains. They all seem to be, for the most part, sorry, I didn't include the T, but for the most part, all of these are known either ketosynthase domains, ketoreductase domains, and acyl carrier protein, which is just the kind of the three canonical protein domains that you see repeated over and over in these modular PKS genes. And so you kind of see those same genes showing up, or the same domains showing up over and over and over again. And then there's some additional kind of enzymatic domains here in addition to those three, but not a lot of disordered or unknown domains of unknown function here. They are generally known protein domains, yeah.
About the hospital,
Yeah.
You know what kind of target.
Say what?
You don't know what is target, like what pathway or what. Why is it alga?
Why is it making this?
Very funny.
Yeah, so there are a couple of hypotheses for why Prymnesium is making the toxin, and I don't think they're really mutually exclusive either. So one, it's using its toxin to kill prey. So the Prymnesium is cytotoxic. It's generally hemolytic. If we incubate the cultures of red blood cells with Prymnesium or just Prymnesium and or lyse products from Prymnesium, the red blood cells will just lyse. So it's not targeting anything specific. It is just it causes cells to lyse. And so what else was I saying? There are maybe the toxin is involved in capturing prey. There is some interesting correlations there with some of our most toxic strains being the most motile. And so you can see if these are using their toxin to hunt prey, they're the ones that are actively swimming versus the ones that aren't using their toxin and are just kind of chilling or not, that it might be something like that. It could also be involved in predator deterrence. So there's some evidence, I don't think from Prymnesium, but in other types of algae that like gill-breathing organisms, like filter feeders or rotifers may come and kind of taste you, a single-celled organism, and if you're toxic, they kind of spit you back out. And so that could be another like cell level benefit for being toxic. Those are the two. Oh, I see a couple of hands going up. So, but yeah, those are the kind of the two main things. And like I said, I don't think they're mutually exclusive. It could be both. Who wants to go? Yeah, go ahead.
Sorry, could you repeat why he had to do it by hand and why the software kept failing?
Yeah, I kind of glossed over it. Okay, there's a couple of reasons. First of all, look at how repetitive these domains are. So these are going to get tagged as repetitive regions of the genome and will get masked. And so when we when you try to predict genes, it will be ignoring these regions because the software will think these are like transposable elements. The other complicating factor here is a lot of times for calling genes, we use transcriptome data. And our transcriptome data that went into our initial gene calls were all poly-A primed. So they were all starting at the 3' end of the sequence. And then we had, reverse transcriptase going that way until it falls off. And there is no way we would ever get poly-A prime data, transcript evidence for the 5' end of this 93 kb protein or gene. And so we only had basically transcript evidence for the 3' end of these two massive gene products. And so that's why I put in parentheses here that this was ribodepleted RNA. So they had to go back and get special RNA-seq data where they didn't polydate primate. They instead just tried to deplete to get rid of the ribosomal RNA. And then they were able to get coverage across the whole gene. Yeah. Yeah.
What about the dark beads? Is it just poisoning and causing the cell to lyse and sucking up nutrients through transporters? Both these cells are thick walled. I can't, how would internalization occur? You know, like phagocytosis is that thought to occur? It takes a minute. Yeah.
Yeah. So that's all the way at the very beginning. Let's play the video again because it's a cool video. Here we go. Prymnesium is phagocytic. That one just has a huge food vacuole. That is a former green alga that has already lysed, and these Prymnesium are phagocytosing up bits of cell debris.
So what's the wall like in this organism?
Oh, so it doesn't have a cell wall, like a plant cell wall. It's covered in polysaccharide scales, and so these scales are kind of overlapping, and a thick layer of mucus. But it is much more of a flexible cell membrane than your typical plant cell. Does that answer your question?
Well, kind of. So then it has to shed all that extracellular material locally, I guess, to engulf things.
Yeah, I don't know if I've seen any like TEM or microscopy looking at Prymnesium in the act of phagocytosing up food material. But yeah, definitely does.
All right. One last question.
Well, so when you look at the genome side of the haploid versus diploid, typically diploid got a much higher double or triple size of the haploid. But I noticed some of the haploid also have a very small genome size, probably even lower than the haploid. That would indicate some of the genome just lost in the evolution. And if that's the case, what are these genome lost? It's the unique sequence or it's duplicated?
Yeah, so again, that if we don't have a diploid to compare 12B1 to, because we don't have a A1 type diploid, we have an A type hybrid that has genes from the A1 lineage and from the A2 lineage. So there could be some A2 type genes that are only present in the hybrid and aren't present in the A1 lineage. So there could be just some presence absence variation due to that. But that's something that actually, stay tuned, because Nathan in my lab is working to get a B type genome well assembled. And so there we really want to look at what's causing genome size variation between A and B types, because if you remember the B type genome, the B type clade, their genomes are more than twice the size, haploid, more than twice the size of a haploid A type. So we don't know, whole genome duplication, I don't think so. I think it's probably maybe like proliferation of transposable elements, but we have to get a good genome assembly and figure it out.
All right, let's thank Jen for a great summary. And I think she can probably stay for a couple of minutes.
Yeah, I'm happy
People
to.
still had a question.
Yeah. Good.